Right.
हाइड उसको नीचे वाले लाइन.
Sure. Yeah. Okay. So, as you know that the topic that I have chosen today is the advancement of artificial intelligence in protein research. So, artificial intelligence is the new buzzword nowadays, and everyone is trying to do something with the artificial intelligence, and everyone seems to think that it's now a one-stop solution to everything or every problem. And of course, whenever a new technology comes, scientists always try to grasp that opportunity and to do something better with that particular technique or that particular technology, so that we can advance our understanding of the different mechanisms or different aspects of the biology. So protein research is one area where artificial intelligence has increased in leaps and bounds, and it has tremendously helped researchers all over the world in different fields, in very different fields, I'd say. In next few slides, I'll give you a brief introduction of what the problem was, what was the research gap, and what was filled by the AI in that particular area. So in the course of next half an hour or so, we'll discuss about the different aspects of AI in different areas of protein research. So I'll start with this. So we'll start with the very basic, which is the central dogma of life. And I guess all of you have learned this one, or at least studied this one in some point of your life. The central dogma, it says that the life starts with the DNA, and DNA being the major molecule, it codes the information for all the biological molecules, and that information is transferred by a transcription to RNA, and RNA then gives rise to proteins by the process of translation, which happens in ribosomes, and the proteins are coded as per the information provided by the DNA. So this directional flow of information from DNA to proteins is known as the central dogma of life. Now, when we talk about the processes of translation and transcription, we always say that the information is coded in DNA and then it is translated into the proteins. But how does that happen? How the information is stored in DNA translates into something that we make the call as proteins. So we know that DNA or RNAs, they are made of nucleotides, and they as per the molecule, if it's DNA, they are called deoxyribonucleotides, and if it's RNA, they are called the ribonucleotides. And there is something called known as genetic code or genetic codons. Now, these codons are responsible for the coded information that DNA carries from generation to generation, and these are the molecules. This is the information which is being carried through the messenger RNA to the ribosomes so that they can synthesize proteins in a specific sequence. So the important thing here is that the proteins has a definitive sequence. So it's not a random alignment or random arrangement of some amino acids, but those the only amino acids which are coded, which they are welded together, which are coded by the DNA. So if you can see this diagram here, it will show you the codons which are present here. So the first layer is the one amino acid, then second and the third amino acid. So we do know that there is degeneracy in the codon, which means that more than one codon can code for a single amino acid. So like you can see, there is a serine here. So UCU will code for serine, UCC will also code for serine. So like this, there is a degeneracy for the codons. Now, based on these codons, the ribosomes, they take up the messenger RNA, and then they summon the transfer RNAs with very specific amino acids bound to them, and then they give rise to a particular protein chain. So this, I'll not go into the details of translation, but this is how the proteins are formed, and the information comes from the codon. So you can see here, this is the codon, this is the anticodon, which is the sequence present on the transfer RNA, and the ribosome makes the peptide bond between two amino acids. So this is how your proteins get formed. Now, why proteins? So all the work that is happening in the cell is done by proteins. if not all, maximum of the work is done by the proteins. So whether it's a structural part, if it's the exoskeleton parts, there are proteins. If it's the enzymes, those are proteins. If there are transporters, those are proteins. If something is involved in movement, like cilia, flagella, those are proteins. Regulations, transcription factors, regulatory factors, the kinases, the dephosphorylases, all are proteins. In communications, the proteins are there. Then in the structure, everything. So majority of your cell is basically proteins. So it's apt to say that the proteins are basically the workhorses of cell. So whenever a cell has to do something, it directs DNA to create a protein, and that protein then transports or it moves to the area where it is needed, and then it does the work that is required by them. So the proteins are the important molecules which we need and which we require to sustain, to live, and to maintain a healthy life. Okay. Now, just look at the properties of the proteins. So proteins, as I told you, are the polymers of amino acids, and this is the structure of amino acid. If you can see this diagram on the right hand side. So amino acid has a chiral carbon center. It has an amide group on the one end. So amide group is this nitrogen accompanied by the two hydrogens, and it has the carboxyl group on the other end, and it has a R group. R group is the side chain molecule, is the side chain which contains some kind of charge, or it's a large chain molecule. So different amino acids have different R groups. The basic being the glycine or the alanine, which does not have the glycine does not have any R group. It has only H here, but the rest of the molecules have different R groups, and depending upon the R groups, they have different properties. So we can so normally 22 types of amino acids are there, and we can categorize them into different categories. Like you can categorize them as polar, you can categorize them as non-polar, or an aromatic amino acids, positively charged, negatively charged amino acids. So depending upon the different properties of R group, you can classify the protein amino acids. Now, when you make proteins from amino acids, you join the amino acids by the amide bonds, the peptide bonds. Okay, so one amino acid, so it joins the carboxyl group from one amino acid and the amide group of another amino acid. They come together, condensation takes place, water molecule is taken out, and a peptide bond, C double bond O NH bond, is formed there, and one amino acid is joined to the another amino acid. Okay, so proteins, as we already discussed, they work as enzymes, structural proteins, hormones, transcription factors, toxins, antitoxins, chaperones, transporters, channels, antimicrobial molecules, etc. Now, there is a kind of structural hierarchy of proteins. So the most definitive aspect of protein is that they have a particular three-dimensional structure. No protein is active without this kind of structure, and they have to undergo a process known as protein folding to achieve that structural hierarchy. So what is this structural hierarchy is? The first is known as the primary structure, which is basically simply the amino acid sequence. This is the linear chain of amino acid sequences, as you can see, and it is known as a primary structure. So it has two ends, the N-terminal end and the C-terminal end. N-terminal end means the amide group is free here. So this is the first amino acid here, and it has the free amide group. And on the C-terminal end, the carboxyl group is free, so that's known as the C-terminal end. And each amino acid is joined by the peptide bond. So you can see this is the linear chain of amino acids and is known as the primary structure. Then there is something called as secondary structures. Secondary structures are the smaller these areas which fold into certain conserved structures which are seen in repeatedly in different proteins like alpha helix. So this is the one domain which is seen as the which has taken the shape of alpha helix. Another one is beta sheets. The difference here is the alpha helix conformation. It comes from I plus 3 the arrangement, which means that the first amino acid makes the bond with the fourth amino acid in the chain, and it forms a kind of compact structure here. Beta sheets are more extended, and their R groups are in the different planes, so they are in the opposite sides. It's a more extended kind of structure here. Then comes the tertiary structure. Tertiary structure is basically the combination of secondary structures. So you can see here you have the beta sheets here, you have the alpha helices here, you have the turns and loops here, and these all combine together to make a tertiary structure of the protein. And sometimes, if your protein has more than one monomer, you need a quaternary structure also, which is basically the active configuration of a protein. So your protein might be active in the monomeric state also, and it can also be active in the tertiary state or in the quaternary structure, where it more than one type of monomers. Either it could be an homomeric structure, it can also be an heteromeric structure also. So more than one type of monomers can also come together to form a quaternary structure. So these are the basic structural steps which protein takes during its folding journey. Okay, so the major or the important aspect here is the protein folding. So the all of these properties of the proteins or the amino acids, they come together to make sure that your protein folds into a particular structure. Now, this protein folding is a physical process by which a linear polypeptide folds into its characteristic and functional three-dimensional structure. So, as you can see, this is the linear structure, and by step it folds and comes together to form this kind of this state here, where it is properly folded and it can actually perform some function. Now, the question comes: why it has to fold into a particular three-dimensional shape? Why cannot it just function like this only, simply remaining as a, as a linear chain? So that question, the answer is, when it comes into three-dimensional state, it creates a kind of space or a cavity where actually the amino acids, specific amino acids, they come together and they form a space where the reactions can take place. So as I told you, the proteins, they also work as enzymes, and enzymes are the biological catalysts. So if you have to take part in a reaction, you have to be able to either donate the electrons or accept the electrons. And that is done by the amino acids. So not all amino acids can participate in the reactions, but only certain amino acids which has the capacity to either donate or accept the electrons or can participate in the bondings can, actively participate in the substrate binding. So those amino acids should come together in a particular three-dimensional space, and the rest of the protein provides a kind of scaffold so that these molecules, they come together and they make a particular that cavity where your substrate or any ligand can bind. Secondly, the protein structures are bound by certain steric rules. These are basically the steric hindrances which creates a problem for their movement. So in this amino acid chain, when all the alpha carbons are here, so there are a lot of side chains are there, and as I told you, all the side chains, they have different properties, they have different charges. So these creates a steric hindrance for the movement of the protein also. So to make sure that all of the molecules are happy in that particular folding, in that particular state, it is also it's important to make sure that the protein has folded completely and appropriately so that it can function. It cannot function just like that. Okay, so how does this protein folding works? So in cell, as we know that in any kind of cell, this process of transcription and translation, they are generally coupled with each other. So the translation begins as soon as the transcription is going on, and with the emergence of mRNA, the protein also starts. The translation also starts, and as soon as the protein starts coming out, the folding, it also starts. Now, if you look at this direction here, this image here, it will show you how the protein folding dynamics takes place. So we have an unfolded protein, and we have a lot of conformations which are possible. So there are a lot of conformations which are possible for a protein to take. Now, because proteins are large molecules, let's say 100 200 amino acid proteins are on an average are there. More than that are also there are large proteins also. And even if a 100 amino acid protein has to fold into a particular state, it can take infinite conformations. But a cell does not have time for that. Okay, so it chooses the best conformation based on the energy of that folding. Okay, so it tries to reduce the global energy of that particular molecule, and it tries to fold it into a structure which has the global minimum energy. So if you have an energy landscape, when you start the folding, it's very broad, lot of conformations, a lot of entropy is there, and then it tries certain combinations, and finally it settles up to a particular state where the energy is low. It's the lowest possible energy for that particular structure. So in this process, you can see an unfolded protein. It gradually folds into a three-dimensional structure here. Now, there is something which is known as the folding bias. So, as I told you, the see cell does not have the time to undergo all the n number of conformations to look at the pattern of how it is folding. So, it has created something called as bias. So, it actually knows which thermodynamical state is more suitable for that particular protein, and it simply tries to adopt that kind of conformation and make sure that your protein is folded immediately, and it does not spend time in rest of the conformations. There are also some proteins like chaperone proteins which help in this process. These chaperone proteins, they act as kind of molds where your the primary amino acid sequences, they undergo this folding process and they assist in the folding process, and then they try to fasten up this process. So this is how the protein folding happens. Also, it's a cooperative folding, which means that if you can fold the secondary structures correctly, they will assist in the correct folding of the another domains. So proteins have some domains, they have some regions which are more easily structured than the rest of them, and if you can structure a domain, it will help in the structure of the next domain. This is cooperative folding, and as I told you, it begins co-translationally. This process is very fast, and it does not wait for the protein to first synthesize completely and then start folding. It starts immediately. Alpha helices and beta sheets are amphipathic. Now, what do we mean by amphipathic is when the protein is folded, there are certain forces which stabilize the protein structure. These are called hydrogen bonds. We have van der Waals interactions. We have hydrophobic interactions. Now, the hydrophobic interactions are very important because many of your amino acids are aliphatic in nature and are hydrophobic. So it is very important because what we are talking about, protein folding or protein synthesis, it all is happening inside cytoplasm. Okay, and it's an aqueous medium. So it has to make sure that the hydrophobic part of the protein or the amino acids which are hydrophobic in nature are not exposed to the aqueous phase. They should be buried inside, and the hydrophilic amino acids should be on the outside of the protein if you're talking about the globular proteins. So the alpha helices and beta sheets are constructed in such a way that they are amphipathic in nature. So their phase, which is exposed outside towards the aqueous medium, it's hydrophilic, and the amphipath or the hydrophobic phase is buried inside of the cell, inside of the protein. So this is how the protein it makes sure that the folding, it should also take into account that your hydrophobic patches are not exposed into the hydrophilic surfaces. Okay, so what are the main players in protein folding? These are hydrogen bonding, van der Waals interactions, hydrophobic effect, steric constraints, and electrostatic interactions. I have discussed a little bit about them. So hydrogen bonding is there, as we know that a lot of hydrogen bonds are formed between the amino acids. In alpha helices, the first amino acid bonds with the fourth amino acid, and so on, and it makes the hydrogen bonds with them. Van der Waals interactions are there. Hydrophobic effect we have already discussed. Steric constraints are because of the large size of the side chains. There is a steric constraint also. So we have to take into account that also, that how much movement can this molecule do. And there is the Ramachandran rules, which are actually which takes center stage here, and which gives us an idea about how much movement can a single amino acid do in a protein or in a polypeptide chain. And then there are electrostatic interactions also. Okay. Now, I think now that we have a little bit idea about the proteins and what proteins are, we can move to the protein structure part. So there is something known as the protein structure determination. So we know by now we have established that proteins have a three-dimensional structure. But how do we know what kind of structure protein has? Because we cannot see the proteins. They are very small molecules. You cannot just see them under the microscope. So you do not know what kind of structure the protein adopts. So we have devised some techniques where you can actually look at the structure, or we can derive at the structure of a protein. The first structure, which was done by the X-ray analysis, was for the myoglobin molecule, which was done by John Kendrew and Max Puritz, and they, received the Nobel for this work. Uh, it at that time, Kendrew, it took him almost, I think, 22 years to obtain the structure of a myoglobin protein, which is only, I think, about 100 amino acids long. Now you can imagine the work that they have done, and the how much effort they have put into, because everything was done manually, every calculation was done manually, and finally they arrived at a structure. And the same thing, if you want to do today, you can do it in a day or two. So, how does the X-ray diffraction actually brings you the structure? So, in 1895, Wilhelm Röntgen, he discovered X-rays. You know that, right? And I think soon after that, the people, they started looking at the diffraction pattern of X-rays with different matter, and the William Henry Bragg and William Lawrence Bragg, this is a father-son duo, they proposed a Bragg's law. The Bragg's law was to find out the diffraction pattern of X-rays over the matter, and they said that if you diffract an X-rays, they diffract by this relation, which is n lambda is equal to 2d sine theta, where lambda is the wavelength of X-rays, d is the spacing between the planes of the atomic lattice, and theta is the angle between the incident ray and the scattering planes. So this gives an idea. So we know because we can control the lambda. So from this we can always find out the angle, that is sine theta, and which gives us an idea at what angle the X-rays are diffracting, or in a particular lattice. So based on this, the technique which was devised is known as the X-ray crystallography. So for X-ray crystallography, what you need is, we need pure proteins. Pure proteins are then crystallized, and this is how the crystals look. These are basically, they look like salt crystals, but they are not actually solid crystals, but they are gel-like substances. You take these crystals, you feed them to X-ray generator. The X-rays are diffracted by these crystals, and the data is recorded on the detection plates like this here, and this data is then fed to different softwares, which will give you an idea about the structure. If data is good, you'll get a good resolution of the protein structure, which structure is then deposited in the protein data bank, which is a public database for all the protein structures. And then you can deduce all kind of applications it have. You can look at the structure function relationship, you can look at the drug designing part, you can look at the discovery of novel protein folds or protein engineering or whatever. It's open to imagination what you can do with that. You can also do NMR. So NMR is the nuclear magnetic resonance, and it is also used for the structural determination of proteins and protein complexes in solution. So here you do not need to make crystals. X-ray crystallography has a severe limitation, is that you need the proteins in crystal form. But for NMR, you do not need the proteins in crystal form. You can use the soluble protein, but the limitation here is large proteins are not easily solved by the NMR. So protein less than 35 kDa you can do with the NMR. So you do the isotope labeling of the sample, generally with the hydrogen isotope. You collect the data and do the structure refinement, structure quality, and if you're lucky, you get a crystal, or you get a protein structure. Then there is something called as cryo-electron microscopy, and this is the newer technique which is now which is used now very frequently nowadays. So basically, cryo-electron microscopy, what you do is, you take the protein, pure protein samples, you freeze them, and you search the grid for the shape of the molecules. So if you freeze the samples like this in this grid here, and you bombard them with electron microscopy, you get this kind of representation of the shape of the molecules. Okay, but now with the increased technology, we have high resolution photographs, and that gives you an holistic idea of how your protein looks like. So cryo-EM works good for large complexes. It does not work on smaller proteins, because then the size of this macromolecule will be very small. But if it's a large complex, larger is better, then it will give you a very good resolution. And we also have the instances where the scientists have solved the atomic resolution protein structure with the cryo-EM. Then there are other techniques also. We have small angle X-ray scattering, we have neutron diffraction, we have circular dichroism. So this is small angle X-ray scattering. It's also performed on the solution, on the soluble proteins. It will also give you an idea about the shape and size of the proteins. It does not give you the high resolution structure, but it will give you an idea about the complex. You have neutron diffraction. Neutron diffraction is done, as you can see, we use neutrons as the electromagnetic radiations. We use the diffraction pattern here. It can also give you good quality data. It can also give you an idea about the shape and the structure of the proteins. Problem is, neutrons, they destroy the crystals very fast, so you have very less amount of time to collect the data. And if you can complete the data in that particular time, you have a structure. If you cannot, then it does not work. Circular dichroism is there. It will show you about the quantity of the other coils, alpha helix, beta sheets in your structures. It will tell you what kind of secondary structures your protein is adopting, and in different conditions, how the composition of those secondary structures is differentiating, or it's changing. That kind of information you can get from circular dichroism, but not the structure. Okay, so the limitations of these techniques are: you need pure protein, of course. You need to first purify the protein. Crystallization is a huge bottleneck. You need to first make sure that your protein crystallizes if you're using X-ray crystallography or neutron diffraction. Data collection is again a tedious process, because data is generally collected on X-ray sources or cryo-EMs. These are very expensive instrumentations, and they do a lot of radiation damage. And if your protein has a lot of disordered regions, it's very difficult to get a quality data. Size of proteins and complexes is also one kind of limitations. Like NMR, it cannot take large amount of data. Cryo-EM, it cannot work on smaller complexes or smaller proteins. So this is also a limitation. Associated cost, as I told you, doing the structure is pretty expensive, and it can take a lot of effort. It can take a lot of resources. So it's not for everybody to do this kind of experiments. Okay. Now, this is something which now comes to the rescue of the scientists. If you cannot determine the structure, can you predict the structure? Can you predict the structure? So, John Kendrew, when he was giving his Nobel Prize speech in 1962, he said that the most remarkable feature of the molecule is its complexity and its lack of symmetry. He was talking about proteins. Now, understand here is that there is no symmetry for the proteins. The arrangement seems to be almost totally lacking in the kind of regularities which one instinctively anticipates, and it is more complicated than has been predicted by any theory of protein structure. Though the detailed principles of construction do not yet emerge, we may hope that they will do so at a later stage of the analysis. Now, you have to understand here is that this is 1962, and they have just finished solving the structure of myoglobin, and they could not look at the patterns, or they could not find the rules or the regulations that they could formulate, and which they can formulate, so that they have a set of rules to form a field which is protein prediction. But as they have predicted that at the later stage there may come some principles which are not yet seen, and that's what has happened with the protein structure prediction. So how do we do the protein structure prediction? Now, there is an inherent problem which is known as the 50-year protein folding problem. Now, what does this say is that the physical folding code, how is the 3D native structure of a protein is determined by the physicochemical properties that are encoded in its one-dimensional amino acid sequence. So the first problem is it possible to deduce the three-dimensional structure of the protein only if you have the sequence information? So if you can answer that question, it means that the information about the folding is actually present in the structure itself. But can we deduce that? Can we determine that just by looking at the sequence? Can we say that okay, this is the protein sequence and this kind of structure it will take? The second is the folding mechanism. A polypeptide chain has an almost unfathomable number of possible conformations. I already discussed this, is that during folding, protein has n number of possibilities which it can take for folding. But how it is possible that the proteins fold so fast? And then, is it possible to predict protein structures using computers? Can we devise a computer algorithm to predict a protein's native structure from its amino acid sequence? Now, we have to remember here is that To do certain work in a in algorithms or in proteins, we have to find some patterns. The algorithms, they work on the patterns. If you can identify a pattern, it can repeat that again and again until it has find out a certain structure. So is it possible to find that kind of pattern in the sequences only? Okay. So here comes the role of the AI and the machine learning. So let me first introduce the artificial intelligence. The artificial intelligence is basically a technique for building systems that mimic human behavior or decision making. We can understand that, right? So artificial intelligence is basically which mimics the human behavior or decision making for us. Then machine learning is basically a subset of AI which uses data to solve tasks. So, if artificial intelligence is about decision making, machine learning is about implementing that decision and to see how it can solve a particular problem. And then comes the deep learning, which is again a subset of machine learning itself, which relies on the multi layered neural networks to solve these tasks. So in deep learning, we automate the task. We say that, okay, this is the data, this is the process, and you have to find out the solution. So that's what the deep learning is. So deep learning, basically the computer, it learns how to perform a certain task. So it does not have to rely on the input. It does not have to rely on always extensively on certain inputs. So it can do it by itself. Now, what is the difference between machine learning and deep learning? So, machine learning, as I already discussed, is the subset of artificial intelligence which involves training algorithms to make predictions or decisions based on the input data. Examples are image recognition, speech recognition, or fraud detections. As you can see, you can use image recognition is used by a lot of softwares like Facebook, and even your cameras have the image recognitions nowadays. Speech recognition is being used in phones and in different different kind of security arrangements. You can use different passwords. So these are all the examples of machine learning. Deep learning is a subset of machine learning that involves training neural networks with multiple layers to recognize patterns in data. Now, these neural networks, they basically mimic the neuron connections that are in your brain, which means that they tend to interact with each other. They tend to crosstalk with each and every layer, and they try to come up with a decision which is most suited to solve that particular task. Okay. So examples are like autonomous driving. So you have heard about the driverless cars which drive by themselves. You have seen the ADAS, which is now coming in more and more cars. So it is basically the driver assisting system. So the data is there because it can look at the road in the front and in the down. It has already been trained how to behave in a certain condition. The data is there, information is there, that what it should do in a particular condition, and when it actually encounters a specific problem, it behaves in a similar way. So if it is going, it sees that okay, the road is empty, there is no vehicular traffic, so it can increase the speed, let's say to 60 or 70. But as soon as it sees that okay, there is a vehicle approaching or a vehicle is going ahead of that car, and it's going slow, then it also reduces its speed. So these kind of decision making, it comes from the deep learning. Okay, so this is what I was talking about. So you can see this is the image recognition. So we give the training data, we give it a data which where we can definitely show is that okay, this is how a cat look like, this is how a dog look like, this is how a bird look like. It does the feature extraction. So deep learning flow is there. It extracts the feature of how a particular organism will look like. It will look at its shape, its distinctive features, and then it learns from that. And when you give it a test data, it will show you that it's a cat. So this is deep learning flow. This is machine learning where you give the input, it feature extracts, it classifies that data based on the input that you have given, and it will give you an output whether it's a car or not. Deep learning, on the other hand, is you give it an input, it will do it the feature extraction itself, it will do the classification itself. There is no need of any human intervention here, and you get an output. Okay, so there is something known as in machine learning, there is something known as supervised learning. So, what is supervised learning? Here, the algorithm is trained on the labeled data. So, this is part of the machine learning. Supervised learning is part of machine learning, and it is basically even your algorithm is trained on a labeled data. So what we are giving we are already know the response, we already know the result, and we are just training the data on that particular labeled here. So the supervised learning models have a baseline understanding of what the correct output value should be. So this model already knows that what the correct answer is. Okay, it just needs to devise a pipeline to achieve at that answer, right? So if you can see this diagram here, so we have a known protein sequence, we have the known responses, we feed it to the supervised learning model, and we let it make a prediction. And then it can, we also tell him that what kind of structure is valid and what kind of structure is not valid. So we let it train on this kind of data, and then when we provide it a new protein sequence, it tries to make sure that based on this information it can predict the structure of that particular protein and can say that whether it's a valid or a non-valid data. Then there is unsupervised learning. In unsupervised learning, the algorithm is trained on unlabeled data. So basically, it does not know what the correct answer is. Okay, so it has to devise, it has to make sure that it understands that what could be the best possible answer with the given data. So we are training it on unlabeled data, and the aim is to identify data patterns or groupings without explicit guidance. It is also required to reduce the bias in the data. So we are not trying to introduce bias into the algorithm so that it can, it can give us a false positive results. So we are giving it data without any labeling, and we are trying to identify the patterns or groupings, and based on the information that is provided in the input data itself. So if you give it a non-protein sequences, supervised learning model is there. Or sorry, this is unsupervised learning model is there, and then it predicts and it tries to make sure that whether it's a valid or invalid data, and then it gives us the result. Then there is reinforced learning. What is reinforced learning is it involves training an agent to work in an environment which reaps optimal results. So agent means the algorithm basically. The agent gathers information from the assessment that is gained in the form of incentives or consequences. So like in the real life, when we make a decision, we also are responsible for the consequences that comes with it, right? So the similar kind of approach we are using with the reinforced learning here is also that we are trying to we are trying to assess a particular decision, and we are also trying to assess the consequences. So we give the rewards in forms of the incentives to the to algorithm, and see whether it's making the right decisions or it's not making the right decision. It kind of mimics the trial and error learning process that humans use to achieve their goals. So it's the reinforced learning. So these are the layers of the deep learning. If you can see here, we have the input. These are the input layers. These are all the hidden layers which you do not see in any kind of program. These are the neural networks which identify the patterns, which identify the combinations, which identify the features, and then there is an output layer, and it selects the best output of that and it says that okay, this picture is of George Washington, and this is how it's happening. So it extracts the features, it looks at all the features that are possible, and then it ultimately tries to come up with the result. This is the deep learning neural network. There are two types of deep learning neural networks. One are the convolutional neural networks, and then are the recurrent neural networks. The convolutional neural networks are structural designs intended for the handling of data that exhibits consistent spatial organization. So you have the data here, then it goes into the multiple layers of convolution. So it has a spatial organization. You can look at that, particularly well suited to image classification and object recognition tasks. It works by transforming an input image into a feature map, which is then processed through multiple convolutional and pooling layers to produce a predicted output. So basically, it extracts the feature, create a map, and that map is filtered through multiple layers, and then goes into the fully connected layers. Here it goes into the classification, and then it gives an output on the based of probabilistic distribution. It says that 70% chance is that this is a zebra, but then there are also the false negatives, like 20% chance is that it's a horse, and 1% chance is that it's a dog. So it depends upon how much your network has been trained and how much data it has in its input. Okay. So what are the advantages of CNN in structure prediction? It looks at the local sequence patterns, are well suited to recognize short range interactions and local sequence features crucial for secondary structure predictions. Similarly, they are also important for translational invariance, which are basically this means that they can identify patterns even when the data is slightly shifted or moved, making them suitable for the flexible and dynamic nature of protein structure. So even if the data is a little bit skewed, it can also make a sense out of that. Then there are recurrent neural networks. So recurrent neural networks is basically which handles the sequential data by retaining past inputs within the sequence though hidden states. So basically in convolutional networks or deep learnings, it's a directional flow. So it all goes in one direction. But in recurrent, it can always come back to the hidden layers, and it can come back and back and until it has reached a significant conclusion. So this is basically when the different layers they cross talk with each other, they come back and they again look at the input data, they look at the various features of the data, and then they try to make a informed guess. Okay, so I'll just skip this. So this is the AI in nutshell. Yeah, this is a very complex picture of AI. You can have supervised learning, unsupervised learning. You have neural networks. You have generative models. Deep learning is there. Machine learning is there. Then ethical AI is there. So lot of avenues are there in for AI. But we are not interested in this. We are only interested in structure prediction. So I'll just move to the structure predictions. So in 2012, Dill et al. Made a statement that similar sequences should yield similar structures. So this became it was already known that there are homology sequences, which means that if you have a protein and you know its structure, and then you look at the sequences which are similar, or have identity of let's say more than 50%, they should adopt the same structure. That's the whole principle of homology modeling. So there is an event known as CASP, which is basically the critical assessment of protein structure predictions, and it started in 1994. This is a biannual event. It happens every 2 years, and here a group of researchers, they are given the data to solve. They are given the protein structure, protein sequences, and they are asked to come up with the best prediction for the structure. Then they try to use different algorithms, they try to come up with different programs, and then they come up with the best software. And if the data it matches the experimental data, they say that okay, this program has worked. So the first CASP in CASP1, which happened in 1994, 33 protein prediction targets were obtained in three prediction categories: comparative modeling, fold recognition or threading, and ab initio folding. So there are three categories basically, and these are the different CASPs. So every 2 years, CASP is happening, and you can see how the target difficulty has increased and the correctness has increased every year. AlphaFold came in I think in 2014, 20. 18 and 20, and you can see the increase in the cumulative percentage of targets here, and the best model achievement also. With the CASP 13 and 14, we have increased the overall quality of the structure. This is the ab initio modeling. You can see from CASP 1 to CASP 14. This is CASP 14 here. So the target difficulty and the overall accuracy both have increased. So the recent advancements have increased our overall understanding of the protein structure, and now the difficult proteins which are difficult for prediction, they are also being solved very nicely by the AI. So, what are the best public CASP certified protein structures prediction servers? We have AlphaFold, we have Rosetta Fold, iTester, Multicom, RaptorX, DMFold, Pcons, HHPred. These are all softwares are available on different web servers. You can use them, you can see them. Then we have the best public CASP certified contact prediction servers like RaptorX, Triplet, Res, Meta, Psychov. We have comparative modeling servers like SysModel and Modeler. We have protein secondary structure prediction servers like PSIPred and JPred. Okay, so how does it started? So it started with the molecular dynamics method, which basically is a biophysical method to understand the folding of the protein. Now, as I told you, there were two approaches to look at. One was the thermodynamic folding, and the other one was the sequential patterns. So initially scientists thought that it's thermodynamics which is driving the protein folding. So they try to look at that kind of features, how it's thermodynamically favorable to fold a protein. So using that approach, they come up with some softwares like Ember, Charm, and Gromos, which are all basically based on the thermodynamics folding. They tried to fold the protein using different conformations, and then they try to find out one which works best, which has the lowest energy possible, and they say that okay, this is equivalent to the native structure of the protein. But the problem with this was it took a lot of time and a lot of computational power, because as we know that thermodynamically protein can take a lot of conformations. So it's not possible for the software to look at each and every state. So even though there were some successes, but not as good as the structural patterns. So a lot of limitations were there. Then came the comparative modeling or homology modeling. So it was pretty easy, and as soon as we have the structure of myoglobin, the another structure was solved for lactalbumin, which has very high homology to the lysozyme. And Greer in 1981 he observed that the structure of a protease could also be divided into structurally conserved regions with strong sequence homology. So he said that even if you can identify some conserved sequences like domains or motifs, and if you can assign the structures to them, it is easier to model that structure on a already known template. So based on this approach, modeler was devised. It was developed by Sally and Blundell. It was the
Model they are
first.
using, sir? Which model are they using?
Sorry.
Previous slide.
Yeah. What were you
Trilokji,
saying?
can you wait for few minutes? Then you can ask question because he's now going to complete.
Right, sir.
Yeah.
Trilokji.
Yeah. So give me just
Yes.
10 minutes, 5 10 minutes
Yes.
more.
Yes.
Yeah. Okay. So modeler is there. Then modeler was it's a homology modeling system, which was it was the first automatic computer program with full atom protein modeling. It used spatial restraints derived from the alignment of the target protein sequence and multiple related structures, and it used this information to model the protein structure. Then you have Swiss model. Swiss model, it came in 1993, and it's basically a pipeline. It's a server modeler. You have to download and you have to use it after installing. Swiss model was a pipeline which was introduced where you can do the comparative modeling. If you know the template, if you do not know, it will search a template for you, and then it will model the structure based on that template. But what if the significant homology is not found? Now, there are a lot of proteins for which you could not find a significant homology of the, because not many structures were there in the PDB. So then scientists came with a process known as threading. Threading is basically a process of aligning a protein sequence with one or more protein structures to look at the folds. So it was basically to recognize a particular fold rather than looking at the whole holistically, looking at a complete structure. We were looking only at some folds. So we know that in nature there are only very limited number of protein structures exist which are known as protein folds, and they can adopt a large number of sequences can adopt into that protein folds. So even if proteins does not have a definitive homology, they can still have a structural similarity. So it's not essential for the proteins to have a sequential similarity, but they can also have something known as the structural similarity. So they can adopt a particular fold, they can adopt a particular conserved domain, conserved motif structure can be there. So the threading approaches have three distinct applications in protein structure prediction: identifying appropriate protein structure templates for modeling a target protein, identifying protein sequences adopting a known protein fold, and then assessing the quality of the protein model. So based on the threading, we have different softwares like iTesser. So iTesser is basically iterative threading assembly refinement. It uses the threading model to generate the protein structures, and iTesser is still working very fine, and it was number one server for protein structure predictions in 7 ACASP 7 8 9 10 11 12 13 14 15, and it not in 13 and 14, but yeah, it was still working better. Then you have the profile based alignments. I'm going to skip that. So now coming to ab initio protein structure prediction. Ab initio means when you have no information available about there is no template available, you have no information about the structure. How can you predict the structure from scratch? That comes under the category of ab initio protein structure prediction. Now here what we do is that the approach starts from the random or open conformations and then simulate the folding process or minimize the conformational energy. Segment assembly based methods are represented by the Rosetta methods, and there are two types of approaches which are used. So we have, sorry, not this one. So we have segment or assembly fragment based. Basically, what you do is you have target sequences. You generate the profile alignments for the different target sequences. So you generate a large amount of multiple sequence alignments. Then it goes into the low phase resolution models using the Monte Carlo fragment assembly. And then you use the physics-based atomic refinement to come at a final atomic model. So this is the assembly of, this is the fragment-based assembly. This is the iTester workflow. Then, okay. Then finally, we move to the AI-driven de novo protein structure. So when we talk about AI, we talk about the AlphaFold. So AlphaFold was introduced in 13th edition of CASP. It's introduced by DeepMind, which is a startup of Google, and it achieved the best position in free modeling, reached a summoned Z score of 52.8 versus 36.6 for the second place. So it's a huge improvement over the score. So what is AlphaFold? It's a machine learning based model for predicting the 3D structure of proteins using only sequence as inputs. It's trained on the known sequences and structures from the protein data bank as well as large database of other protein sequences. So how does it work? You have the input sequences. It takes the input sequences, it generates a multiple sequence alignment. Now, when it's looking at the multiple sequence alignment, the simplest explanation is it is looking at the position of amino acids and the propensity of the amino acids at what kind of structure they will adopt in that kind of situation. So that information it is trying to extract from the multiple sequence alignment. And when we say multiple sequence alignment, this sequence alignment can be in thousands of sequences. So it can generate a large amount of multiple sequence alignments, and from that it will derive a consensus information that a particular amino acid has a large probability of adopting a particular conformation in that particular amino acid sequence. So that information it is taking, it's going into something called as evoformer, which has 48 blocks. Then this information is taken into the structural module, and then at the end you have a structure which is modeled on the base of the high confidence and low confidence. The portion which has the high confidence is modeled in different color. The portion which it cannot model with the very high confidence is shown in like this here, which is basically in the low confidence here. It also recycles these structures 3 times from this pipeline to arrive at a particular model which is more and more accurate with respect to the native form of the protein structure. This is the AlphaFold architecture. So you have an input module where you input the protein sequences. The protein sequences are fed into the multiple sequence alignment. So it generates a multiple sequence alignment. It tries to find the structural templates if it could. And the structural templates does not mean the homology templates, but these are basically the templates for these amino acid motifs and residues or the patterns if it could find. Then it goes into the evoformer. Evoformer is basically the learning blocks. It goes into this layer here. The structural modules are formed. Then this is the backend decoding. This is the calculation that it is doing at the backend, and this is then decoded. There is a decoder module which decodes this information into the structural information here. So it tries to extract the spatial information from the multiple sequence alignment and the structural modules, that a particular amino acid, what kind of spatial position it could take, what kind of structure it could take, and then it tries to apply that information into a holistically complete structure model. The accuracy, as you can see, has increased from 30% to almost 80-90%. It's not touching with AlphaFold 3. It's more and more it's coming. This is the scale of goodness. This is the prediction of how good your structure is. There is a score called as PLDTE, which is given by the AlphaFold models, and PLDTE is basically the predicted local distance difference test, and it is scaled from 0 to 100 with higher scores indicating higher confidentiality and usually a more accurate predictions. This is the web server of AlphaFold protein structure data. Actually go and look at the structures here. This is the one structure that has been solved by the AlphaFold. This is a potential plant disease resistant protein, and you can see this is a complex protein structure that was modeled completely by AlphaFold without any kind of structural resemblance. Now the recent one is AlphaFold 3. It has replaced the AlphaFold 2 evoformer with the simpler pair former module. It directly predicts the raw atom coordinates with the diffusion model, replacing the structural module that operated on an amino acid specific frames and side chain torsion angles. This is the overall effectivity of how much advancement the AlphaFold 3 has made. Now we can actually model the DNA and protein predictions. You can look at the interactions of DNA and protein itself. You can look at the human coronaviruses and the heavily glycosylated proteins also. So the modified proteins can also be modeled with this. These are the people who are behind the AlphaFold. These three people have recently, got Nobel Prize. So the applications for AlphaFold are, we can do the molecular replacement for experimental crystal structures. You can do the ligand binding and cofactors. You can do multi-domain modeling using AlphaFold 3. You can do protein nucleic acid complexes using 3 and Rosetta Fold Na. You can modeling orphan proteins using ASM Fold and Omega Fold. So the major part is the actually the drug discovery and design. So AI can be used for drug discovery. You can use it for target identification. You can use it for virtual screening. You can use it for structure activity relationship modeling, de novo drug design, optimization of drug candidates, and drug repurposing. So this is a huge boon for the people who are involved in the drug discovery process, because we know that drug discovery takes a lot of time, it takes a lot of effort, and with the predictive softwares that we have now, the predictive technology that we have now, we can actually filter out a lot of molecules, and we can actually predict a very good binders, which will make more better candidates, which are suited better for the ligand purposes. And that saves a lot of cost, saves a lot of effort. And this is the one achievement of AI that we can actually commend. This is the drug discovery pipelines. You can see I have also listed some success stories, but I think I have already gone up with my time.
Ah,
I'll stop here. Yeah, so I'll stop here. And the one final thing is that we thank all the people who are basically providing structures to the PDB, without which nothing would have been possible. So I'll stop with this.